Hi, my name is Stephanie, and today we are going to talk about DNA, proteins, and mutations. So let's say we have a protein that is 4 amino acids long that is formed from this DNA sequence in vivo. We are able to label the top DNA strand as the coding strand and the bottom DNA strand as the template strand. During transcription, all of the T's from the coding strand are replaced with U's, which will give us the following strand of mRNA. We can then use this strand of mRNA in translation along with the handy-dandy coding dictionary to determine the amino acids in the protein. The start and stop codon are highlighted. There are 4 types of point mutations we will talk about today: missense, nonsense, silence, and read-through mutations. But before we talk about mutations, we need to understand the difference between transition and transversion. A transition is when a purine mutates into a purine or a pyrimidine mutates into a pyrimidine. And transversion is when a purine mutates into a pyrimidine or vice versa. A missense mutation changes the amino acid that is coded for. To the right we have a normal and mutant strand of DNA. After transcription, we get a strand of mRNA with the mutation. After translation, we get the strand of amino acids. The missense mutation changed the amino acid tryptophan to arginine. In this specific example, the mutation was a transition mutation. A nonsense mutation changes an original codon into a stop codon. To the right, we have a normal and mutant strand of DNA. After transcription, we get a strand of mRNA with the mutation. After translation, we get the strand of amino acids. The nonsense mutation changed the amino acid tryptophan into a premature stop codon. In this specific example, the mutation was a transition mutation. A silent mutation is when the mutated codon codes for the same amino acid as the original codon. To the right, we have a normal and mutant strand of DNA. After transcription, we get a strand of mRNA with the mutation. After translation, we get the strand of amino acids. While the silent mutation did change the codon, it did not change the amino acid. In this specific example, the mutation was a transversion mutation. A read-through mutation is when a stop codon is changed into a codon for an amino acid, resulting in a longer protein. To the right, we have a normal and mutant strand of DNA. After transcription, we get a strand of mRNA with the mutation. After translation, we get the strand of amino acids. The read-through mutation changed the stop codon into an amino acid, tryptophan. In this specific example, the mutation was a transition mutation. Thank you so much for watching.